All right, good morning, everyone. Welcome to our third session of the RMP Summer Series. Today, I have the pleasure of introducing our first international speaker, Dr. Mohamed Abazeed. So Dr. Abazeed is a radiation oncologist and associate professor at Northwestern University's Feinberg School of Medicine. He's a clinician scientist with a focus on the use of trans-omic approaches to improve the personalization of cancer therapies. He received his MD and PhD degrees from the University of Michigan with a research focus in model system genetics. Dr. Abazeed is the scientific director and the co-leader of the lung cancer program in the Lurie Cancer Center. He's also the inaugural director of the Center for Precision Radiotherapy in the Department of Radiation Oncology. So please join me in welcoming Dr. Abazeed this morning.
Thanks so much, Michael, and it's certainly a privilege to be back here. This is my third trip to Toronto, visiting Princess Margaret, so I hear two more and I get a set of steak knives. Okay, let's talk about finding signals amidst all the noise. I'm really excited to talk to a group like this. I know you guys are heavily invested in understanding genetic determinants of radiation sensitivity, and you have a very robust translational program, so I'm looking forward especially to the Q&A session. So first, my disclosures. I think most of these are going to be relevant here, so I'll give you more than a millisecond to look at them. Oh, of course. Okay, so what is our research program? Our research program is effectively trying to personalize radiotherapy. I understand that's a very vague statement. And one could consider us the ultimate in personalization already, using CT-based, anatomically selected features, contouring tumors on individual patients. So in many ways, we're already personalized. But I guess what I mean is the use of biomarkers to potentially direct patient care. This is something our medical oncology colleagues have done a very good job of. We've had more challenges, and I think you're going to begin to see why those challenges are not just trivial, they're actually pretty important. And so the right treatment for the right patient at the right time, effectively, you would identify a biomarker, a diagnostic. This could either be transcriptomic, genomic, epigenomic, or even image-based. And we'll get a chance to touch on the image-based features towards the end here. And then the idea is to bin your population. This is obviously a drug here, but to be able to select patients on the basis of what dose of radiation to give them. So we think a lot about prediction modeling in radiation oncology. What could this look like? So in medical oncology, of course, it's a binary classifier. We don't really distinguish between different types of EGFR mutations or different types of ALK translocations. We tend to bin them as either yes or no, and then give a drug. However, there are different types of EGFR mutations, and they confer different levels of activity in EGFR. Also in KRAS, there's tremendous diversity in the specific mutations in KRAS, for example. We'll talk a little bit about that shortly. And so it's very useful information that we're discarding that our therapy, I think, can be uniquely applied for, which is we don't have to either give radiation yes or no. We can give radiation in increments of fine doses, even decimals if we wanted to, although we tend not to do that. And so I think capturing that type of nuance in your biomarker as a continuous variant can be really helpful, especially for the radiation oncologist. The other important thing to consider is time to event. And so if we were going to dose escalate and the patient has a probability of surviving only 2 months, and the predicted local failure isn't going to occur until 10 months later, then perhaps that's not the right patient to dose escalate on. So we'd want to be able to not just predict whether local failures occur at 5 years, 10 years, 2 years. We'd like to know when they're going to occur. So you want a time-dependent measure of the outcome. I think all of us can agree that that is quite useful if you can get that information. And so what is this theoretically magical paradigm going to look like? You can stratify patients on the basis of various genetic and non-genetic biomarkers. You can assess their potential risk of a bad outcome, local failure, and then you can potentially adjust the dose of radiation to mitigate that bad outcome. Of course, if some in the audience are already thinking there's the competing risk of non-irradiated field recurrence and so on and so forth. But I think we all, at least on some level, ascribe to the Hostidian hypothesis that local control is very, very meaningful. And especially in particular sites like head and neck cancer, I think it means a lot. And so we think this is useful information. The challenge is pretty daunting. This isn't a single gene, single drug phenomenon. Radiation is much more complex than EGFR targeted therapies or ALK targeted therapies. First of all, the probabilistic trajectories of ionization tracks are probabilistic, they're random. It may depend on whether you affect a cell kill, whether you have the right orientation of the DNA, and whether you can create enough clustered damage to incur death. And so how on earth are you going to be able to make predictions in this space? In addition, tumors are really complex. There's lots of gene regulatory networks that are activated or inactivated depending on the specific mutations in cells. Medical oncologists, again, really depend on progression-free survival. So if they can push the tumor back by a few months, that is a useful biomarker. For us, local failure is a pretty stringent criteria. Perhaps we need to think about reevaluating that, but for now, that's the criteria. And so if there are a few cells that happen to stochastically survive, despite our best prediction models telling us that that's a very ultra-sensitive tumor, so by the way, this is the butterfly effect of Lorentz transformation, then those stochastic survivors will lead to recurrence, making it way more challenging to be able to predict responses to radiation. Of course, there's been a lot of debate, intrinsic versus extrinsic determinants of radiation sensitivity. The radiation oncologists have had their say for the last 10 years. I think the tide is turning. It doesn't seem like it's that easy. You don't simply irradiate tumors and activate an immune response, but nonetheless, we need to account for other components, including a tumor-associated fibroblast, as well as the immune repertoire. And we'll talk about how to potentially model that in human tumors. And then lastly, these tumors are dynamic. We typically get a biopsy at the time of diagnosis. We have the salient genetic features of that tumor at the time of diagnosis. However, they're undergoing constant evolution, especially after you apply a stressor. This is actually a head and neck tumor. This is a cartoon version of what actually happened to a head and neck tumor. This is a subclone that emerged, and this is the phenomenon in complex system called spontaneous order, where this tumor effectively looks like a minor component of its original self. So how do we begin to even understand this complicated process? Well, what my lab has been doing for the last 10 years is building scaffolds, useful translational systems to be able to interrogate these different features. And we think, although all model systems are imperfect in some way, we think these scaffolds, especially the more advanced patient-derived xenograft models, get us closer to the human tumor in its original place in situ. So this is roughly what this effort has looked like. We've been collecting surgery and biopsy samples from thoracic patients for over 10 years. We've been collecting blood. These undergo various types of profiling. You'll see some of that later in terms of the human tissue collection, the DNA, RNA. Protein we haven't done so much on, although we've been more interested in protein and metabolomics more recently. For the vast majority of these, the original intent was really to get a living biobank. So what does that mean? That means taking a human tumor, injecting it into the flank of granted an immunocompromised mouse, but it still retains some of the immune environment in which it originated. And then developing ex vivo systems that are more tractable for genetic manipulation, and you'll see examples of that. On the other side is collecting, this is an integrated network. So what does that mean? That means we have extensive information, not just about the tissue, but also about the patient and the treatments that they underwent. And so extensive histopathology, EMR, so whatever outcome you're interested in, local failure, progression-free survival, overall survival, tobacco history, and so on and so forth. And then in the last 5 years, I would say we're really interested also in imaging analysis to incorporate into this alchemist-like model system that can theoretically and magically predict the outcome. Okay? You should be skeptical. So the original effort where this all started was as a fellow at the Broad Institute, Dana Farber and Matt Meyerson's group. We had access to the Cancer Cell Line Encyclopedia. CDD Squared was assessing drug response to this Cancer Cell Line Encyclopedia. So we begged Stuart Stradburn and Matt to give us some of the cells for radiation profiling, and we were able to profile 533 out of the 900 cell lines. A lot of those were leukemia and lymphoma cell lines that we didn't profile, and some failed QA. And so this was published back in 2016. We're going to cover this very, very briefly, only in terms of the methodology. More recently, we culled the single nucleotide variant information, the mutation information from this profiling effort, and in a directed fashion, in a very laborious arrayed fashion, we tested the individual effect of these variants, 400 in a immortalized epithelial lung line. And this was published recently in 2002 in Clinical Cancer Research, and we'll go over briefly this effort. PDX-ray is a project that really should stimulate me getting my head examined. This has been our purchase of Twitter, our deserving of a Purple Heart, hopefully not because we're killed in action, but because we're only heavily wounded. This has been a very costly and laborious project. 500 PDXs, 10,000 mice, irradiated at various doses, and we've encountered every possible problem you can think of when you imagine such a project, but we're 50% of the way through, and you'll get a short update on that. Okay, so this all started with miniaturization of the colony formation assay. I think most of you are familiar with this. I'll quickly gloss over the salient features here. We developed a high content assay using initially a 384 well format, and then modified it for 96 well format. What we saw was there was tremendous intrinsic differences in the sensitivity of cancer cell lines to radiation sensitivity. So you had a phenotype, resistance and sensitivity, a continuous variant, and we use information theory to associate the multiple genetic features that were cataloged in the cancer cell line encyclopedia with this phenotype. So you have genes that were associated with, not causing, but associated with resistance and associated with sensitivity. So because of that mere association, obviously we picked a few vignettes and verified that those important genes were in fact conferring resistance or conferring sensitivity. We wanted to do this in a massively parallel way. And so the cancer cell lines are obviously genetically complex, and so it's hard to tease out particular mutations even when they're heavily associated with the phenotype. So effectively what you want to do is take that mutation, pull it away from that complexity, and put it in a cleaner genetic background, in an isogenic background. And so for that, we picked the BS2B cells. These are immortalized cell lines, so SV40 immortalized cell lines. So they lack a little bit of P53 and they lack a little bit of RB because of the SV40 antigen, but otherwise no oncogenic driver, no other somatic mutations of relevance. And so we took the information from the profiling effort and we classified variants on whether they conferred resistance or sensitivity. We did a couple of computational things here. A gene could either have a mutation or not, so we label that as 0 or 1. And if it had a mutation in a structured part of the protein, this was really important for picking out variants that conferred resistance to radiation, because typically, generally, not always the case, but typically when you have a resistant allele, it occurs in a driver, and it typically occurs in a structured part of the gene. It's activating something, right? And those activation events typically occur in structured parts of the UniProt. So UniProt is a big database of proteins that tell us whether there are domains there or not. So this was really, really important for enriching for resistance alleles. This is information theory. It effectively looks for associations. It's not so dependent on linear effects. So you can have nonlinear effects. If there's an association, it's a highly sensitive way of detecting that information. And so ultimately, we had a big list of variants across 92 genes, many, many variants, and we picked the top genes and we assess them for radiation. The way we did this was, I keep moving this. The way we did this was if based on computational analysis, if the gene was activating, if it was an oncogenic driver, you simply put that gene in despite the 2 copies in this immortalized cell line. So there's endogenous as a diploid cell line. However, if there was loss, thank you, whoever did that. Awesome. However, if there was a loss of function allele, of course, that's not going to work unless it's a dominant negative. You have to eliminate the endogenous 2 copies. So, necessity being the mother of innovation, we devised a way to be able to do this and still use our extensive SNP collection, single nucleotide variant collection. This is actually a really cool way. If anybody does CRISPR, I highly recommend you do this, even in cases in which you don't want to add back your gene of interest, which is targeting the intron-exon junction of a gene. So remember, we're introducing a transviral gene, an open reading frame of the gene, and you need to inactivate the 2 copies and yet have this resistant to your CRISPR guide. And so by targeting the intron-exon junction, the ORF is resistant to that guide, and it's actually substantially more effective at knocking out the gene of interest. So we actually now do this even when we just want to eliminate a gene in cells. And so there's a few online resources. You can look at our paper to find those online resources to pick the best guides for this intron-exon junction approach. And obviously you want to target early on in the gene so that you can have full deletion or almost full deletion of the gene of interest. So same high content platform, you make these changes, you infect with virus, and then in an arrayed format, you're able to assess radiation sensitivity, and you're able to catalog, whether you knock out the gene of interest or not, you can now catalog gain of function, loss of function, dominant, negative, and so on. So this is what this looks like. So first, let me orient you here. Every run, this took us 24 months to complete. Every run had a NRF2 E79K. We're going to talk about NRF2 at the request of your fearless leader here extensively. So we're going to get to that shortly. So this gave us one of the strongest phenotypes in our original profiling assay. So we use this to benchmark resistance, and we knocked out 53 BP1. And so these were included. So this is the range of noise around each of these across 112 runs. And so what you're looking at, effectively anything above 1 standard deviation, we consider to be of interest. You can see some NRF2 alleles come to the top, some KEAP1 alleles come to the top, some KRAS. This is arranged alphabetically. But you can see the who's who of signaling pathways of potential oncogenic drivers. So what did we learn? First, a really great sanity check. So each of these variants were culled from cancer cell lines. Each cancer cell line had an area under the curve, a radiation sensitivity characterization that was done in 2016. I told you about that. And now we have taking that SNV out, and we have phenotypic data on that SNV individually. So if you compare the average AUC of the cancer cell lines binned into quartiles with the SNV log 2 AUC, so resistance here, you see a graded response. So that's exactly what you hope to see. So generally, if you take SNVs from highly resistant cancer cell lines and you put them in the immortalized background, they tend to give more, not always, but generally they tend to give more resistance than SNVs that you took from sensitive cancer cell lines. So that's cool, right? Because it suggests that these main genes, that this computational approach is enriching for the most relevant SNVs that you have in these cancer cell lines. So there's a bunch of computational tools. A lot of people say, well, why on earth are you doing this? Just look at the conserved residues in proteins. There's already some characterization of some of these alleles. Just rely on computational tools to predict salient or phenotypically relevant alleles. It's because that doesn't always pan out. So there's a couple of examples of oncogenic drivers, for example, that conferred radiation sensitivity. Here's a huge list of variants of unknown significance that we profile. So some of these in really important genes like RHO-A and KEAP1, for example. KEAP1 is the cognate binding partner of NRF2. So E117K is by all intents and purposes a variant of unknown significance. Some have suggested that it's even neutral. It has no phenotypic consequences. But in our assay, it gave a really strong signal for radiation resistance. So clearly, these types of bespoke phenotypic assays, especially for your phenotype of interest for radiation, which hasn't been done before, can give you important information for variants even computationally may be incorrect. And so here's the validation of both RHO-A. So this is really interesting. RHO-A has canonical mutations that activate the gene. F106L isn't one of those. Now I can assure you that it is one of those. And so we're helping the medical oncologist characterize these functional mutations. And so once you reintroduce this, you can see significant resistance with both this RHO-A allele as well as this KEAP1 dominant negative allele. So this is our CRISPR strategy. I'm going to talk a lot about KEAP1, and I thought this would be a good introduction into this particular gene. So KEAP1 is really complicated. So KEAP1 binds to NRF2. If you degrade KEAP1 or delete KEAP1, NRF2 is free to do its thing. NRF2 is a transcription factor. It goes into the nucleus and activates a ton of antioxidant response elements. So these are our enemies as radiation oncologists because they can detoxify free radicals. So this is a pathway mostly associated with tobacco consumers. Tobacco consumption leads to oxidative stress. So cancers have figured out a way to activate this pathway to allow tobacco-associated tumors from growing despite the oxidative stress. So obviously of real interest to the radiation oncologist. So you can delete, so this is the exon intron approach. It creates an indel in this particular monoclone characterized several monoclones that lead to a similar phenotype. So at least a deletion here, and you get a skipping effect, and you get loss of KEAP1. And then it's resistant to the guide, as I'll show you here. So KEAP1 is actually really expressed at really low levels. And so you don't need much of it to degrade NRF2. NRF2 itself is expressed at really low levels. So these are kind of difficult questions to run. When you delete KEAP1, you see loss of the protein, and you see significant resistance to radiation. Okay, so you then put in an M-cherry or vector control, no effect on the radiation phenotype. When you put wild type KEAP1, you can see here, in this case, we put it under a highly expressing promoter. You see it as restoration, stable restoration of the KEAP1 protein and the restoration of sensitivity. So we can play with the system. We can make cells more resistant and bring them back, which is really cool. And I'm going to show you another application of that shortly. So this is NTC. So this is this control. And you can see here that some alleles of KEAP1 are dominant negative, right? Because they still have the 2 copies of KEAP1. You put in those alleles, they confer radiation resistance. However, when you delete KEAP1 is really when you begin to see the loss of functional alleles. So this is critical. The fact that you're deleting the other 2 copies is critical for phenotyping KEAP1. The other thing I want to point out here is this isn't a binary phenomenon, right? KEAP1 has multiple couch domains, multiple interacting surfaces with NRF2. And not every allele is created equal. Not every allele has the same effect on NRF2 activation. Some really activate NRF2 and some barely activate NRF2. So again, this continuous response and thinking of mutations as binary, is a gene mutated, yes or no, and is that going to be by biomarker? Hopefully, if this paper has done anything, is dispel that myth in radiation oncology, at least. So what do the vast majority of these oncogenic drivers do? They rewire. They do many different things, and it depends. So it depends on what pathway you're speaking about. But generally, driven by KEAP1 and NRF2, they rewire the metabolism. And so, as experts in hypoxia and oxidative stress, perhaps not so surprising, they effectively increase. These are the transporters that are activated. Increase glutamine, cysteine uptake. Increase gene expression of molecules important for glutathione synthesis. And glutathione reductase. And if you deplete some of these cells, so this is an NRAS Q61 allele that was highly resistant to radiation, Q61K. If you deplete it of cysteine and then restore it back, you can see this radiosensitization effect. So it appears that at least NRF2, KEAP1, and some RASs do a really good job of rewiring the metabolome. So that's causality, that's biological possibility. So let's talk about NRF2 and KEAP1. The provenance of these findings is really complicated. There's many people working on these pathways. So the field starts off in the 1970s. We're not going to go that far back, but I want to give the toxicology researchers their due. They've been studying this pathway in a non-mutation, non-cancer setting for over 4 decades. So NRF2 KEAP1 is really important in liver metabolism. It gets transiently activated in the liver and can detoxify drugs or poisons that we take in. However, when it comes to cancer-related mutations in NRF2, this was a really, for me, a canonical paper by Shibata et al. PNAS that identified specific mutations in 2 domains in NRF2 that mediated binding to KEAP1 and therefore lead to activation of NRF2. The first group is to show NRF2 mutations and KEAP1 loss confers resistance to radiation is Shine Biswell's group at Johns Hopkins. Okay, that's the provenance of the finding. They found in 2 cancer cell lines, A549 and H460, KEAP1 loss of function alleles and KEAP1 loss that led to radiation resistance. And they did the complementation work back to be able to assess that. When I was a fellow, in addition to developing this assay, Matt Meyerson was in charge of lung adenocarcinoma and lung squamous profiling for TCGA. And Peter Hammerman, who was the postdoc at the time, working in lung squamous cancer, found an inordinately high rate, previously unappreciated rate of mutations of NRF2 KEAP1 in lung squamous cancers, right? Heavily tobacco associated, like head and neck tumors. So high rates of NRF2. And Matt and Peter said, hey, Mohamed, this might be interesting, oxidative stress response. Why don't you look at this in the lung squamous lines? So we took CCLE, characterized the lung squamous lines, and showed that this was the main pathway of resistance. There were p53 and other minor pathways, but this was the main pathway of resistance in lung squamous cancers. We then showed KEAP1 mutations in our 2016 paper were heavily associated with radiation resistance in lung adenocarcinomas. So there's slight differences in the rate of KEAP1 NERV2 mutation we'll get to shortly. This is a paper by Thalas in collaboration with Tyler Jacks, developed a mouse model, genetically engineered mouse model of KEAP1 loss, and showed that this metabolic rewiring dependence on glutaminolysis in KRAS-driven lung cancers. We're going to talk a little bit about this shortly. Max Dean comes along and then publishes two cool papers. The first one effectively shows what Shine and we showed in lung squamous cancers, but uses a nice orthotopic mouse model to show that. And then in addition, his next paper looks at a clinical cohort, and that's what we're going to talk about. And then finally, the massive phenotyping of KEAP1 alleles that we just saw is in this last paper. So we by no means have saturated KEAP1. KEAP1 is 585 residues. There's a ton of residues that have been saturated, but those are the most alleles that have been profiled for KEAP1 to date. All right, so we talked a little bit about this. NERV2 through these two motifs binds to KEAP1. If you have mutations in either this interface or in NERV2, it leads to activation of the gene. Badness happens because it activates antioxidant enzymes, allows the tumors to grow a little bit faster and become more resistant to radiation. The rates are really high in lung squamous cancers. 34% of the altered samples had either mutations in NERV2, KEAP1, or the choline ligase COL3. And so in adenocarcinomas, 20% mutation rate in KEAP1, vast majority of them are in KEAP1, a small percentage are NERV2. So almost never see NERV2 mutations in lung adenocarcinoma, which is really interesting. Why does that happen? And what is the mutational process that leads to those differences? So this is Max's cancer discovery paper from 2020. He took a cohort of chemoradiation treated patients and characterized them for NERV2 KEAP1 mutations. The numbers here are limited. I think pretty on brand. So take it with a grain of salt. I think you have to be careful if one of these mutations skips categories. I think the significance here can be quite trivial, but you'll see there's accumulating evidence of the importance of this pathway. So SBRT cohort and chemoradiation cohort and a surgical cohort, which is really nice control. In a surgical cohort, KEAP1 NERV2, you don't care, you're removing the tumor. It's not, who cares about oxidative stress? There's no effect. However, in the radiation cohorts and the chemoradiation cohorts, you see these mutations confer resistance or higher local failure rates to after radiation. To Max's credit, he points this out. Look, there are other features. There's clinical features, there's imaging features. Here, volume is a determinant of recurrence as well. So it's important that we think about integrating these biomarkers in addition to other known clinical variants. For SBRT, for example, tumor volume is really the best predictor of local failure. The more clonogens you have, the more likely that those clonogens are going to survive and recur. This is another cohort. This is a PORT cohort, postoperative radiation. And so this is a little bit more complicated. 16 patients, they grouped together these patients in a deleterious or resistant collection of mutations. STK11 is frequently co-mutated with KEAP1. We are going to talk about that, especially the overlap between KEAP1 and STK11, and then NERV2 and PIK3CA. So in this cohort, if you pick these really bad genes out, they tend to recur within the irradiated field at higher rates. There's also a definitive chemoradiation with and without Nivo. There's a suggestion that Nivo suppresses the rate of local failure, but KEAP1 NERV2 mutations are thought to be great candidates as potential biomarkers of local failure in both lung cancer as well as tobacco-associated head and neck cancer. So how do we target these mutations? There's been several attempts. The initial four, unfortunately, have been unsuccessful. So this is CD839, which is made by Calithera, telangestad. And so this is blocks GLS1, so target the metabolic vulnerability of KEAP1 NERV2 mutations. As single agents, he's had very little activity. And so unfortunately, Calithera stopped development of CD839. And we were actually hoping, I think Ben was on the call, we were hoping to get CTEP to give us approval to test this with radiation. Unfortunately, that drug isn't going to undergo further development. There are additional drugs that are in the pipeline that will be interesting, but effectively the strategy here is these tumors want to make more ROS metabolizing substrates, and so prevent them from doing that either by targeting the upstream or downstream effects on metabolism. We have other strategies as well in an ongoing collaboration with Bayer. We have shown that high LET radiation is very effective at killing these types of tumors. So just remind you, this is sparsely ionizing radiation, cluster damage, high LET or alpha particle radiation effectively creates a concentrated ionization tract that is more lethal. So the RBE is estimated to be about 8 to 10 compared to sparsely ionizing radiation. And since it's most of the effect of alpha particles is direct rather than indirect, potentially they might not care so much about KEAP1 NRF2 mutated tumors. So we had developed this assay, sponsored research with Bayer, in which we can measure the effects of radium disintegrating into its multiple daughters, and we can actually develop a nice microdosimetry scheme. And here are NRF2 KEAP1 mutations, and you can see radium just flattens them, effectively destroys them. We took the same BS2B cells and we put in a NRF2 mutation. You can see for cesium, which is sparsely ionized radiation, the NRF2 mutations confer lots of radiation resistance. However, for radium, these curves collapse. So if you land an alpha particle on a lung tumor with a NRF2 KEAP1 mutation, perhaps this resistance won't be observed. And so we're now developing strontium. Thorium is less in favor now because it degrades into radium and eventually goes to bone. So we're developing strontium-based ligands, and I can't tell you about the targets, otherwise Bayer will kill me. All right, so how about we repurpose this information for other cool uses like radiation protection? But unfortunately, you can't just delete KEAP1 because it's oncogenic, or at least it's involved in a process called permissive oncogenesis. So you need another driver. You can't just delete KEAP1, you don't form tumors, but if you have another driver, so you have patient who's 70 years old has a in situ carcinoma that's KRAS driven, and you delete KEAP1, that's really bad. You're going to drive that tumor. So how do you reversibly delete KEAP1? This is our approach. Love your feedback on this because we're in the early stages of this. So we don't want autonomous, we want user control of manipulating the genome. And this is a little bit abstract, granted at first. We're doing this in cells. Eventually, I'd like to take this to cell therapies and then potentially in mice. So the model here is that use a drug to switch genes on and off, right? And we're going to use established methodologies for activating and inactivating genes. For KEAP1, I think is really attractive target from a radiation protection standpoint because you're removing a product, which tends to be really quite easy. And so you can do what's called detagging. You can use multiple ligands here, but effectively, if you target the two loci of KEAP1 in your immortalized cell line, with this DTAG, you can add a drug, a molecule that selectively degrades that protein. And these DTAG compounds are being used in multiple myeloma, so you know that they're safe for human use. And so we actually did this. We did this in BS2B. We inserted a fusion protein at the ends of both copies of KEAP1 in our immortalized cell line. And when we add the product disappears. The drug KEAP1 disappears. And when you remove the drug, KEAP1 comes back. Sorry, I don't have that for you. I should have included that. But upon drug incubation, you see significant resistance, and when you remove the drug, it restores the activity of KEAP1. There are ways of activating other alleles, and so this is kind of a cool experiment to be able to manipulate cells. Where could this potentially be applied? I think it can be applied to CAR-Ts. So as some of you may know, CAR-Ts undergo extensive genetic modification before being infused in patients. In fact, so not only are you activating a particular receptor, but you're also knocking out a ton of things to prevent allogeneic refusal or graft versus host disease. And so we think that this could potentially be a tractable strategy to make T cells selectively lose KEAP1 by adding a drug at the time right before radiation. Now it's a relative contraindication to radiate patients using external beam radiation after infusion with CAR T cells. And so our next step is, after completing the work in the immortalized cell line, we're going to go to the CAR Ts and see if we can affect this phenomenon and see the fold resistance to lymphocytes that we can induce. There are other genes that are really important beyond KEAP1. So we wanted to test the interaction. So when you look at human cancers, both KEAP1 and KRAS are co-mutated at significant rates, and so are KRAS and STK11. So the cancers have decided to co-mutate these pathways. We tend to believe the cancers and that these pathways are important. So we thought potentially if you were to create mutations in both KEAP1 and STK11, that would lead to potentially a stronger phenotype. And so that's exactly what you see. STK11, which is involved, another molecule that regulates metabolism. Nothing happens when you delete STK11. KEAP1 deletion, I've already shown you, causes resistance, but when you do the double knockout, you see a significant increase in radiation resistance across two clones. The same for KRAS, by the way. So KRAS mutations, multiple KRAS mutations were tested with and without STK11 deletion. And what you see is substantial increase in radiation resistance when you knock out STK11. So we think not only can you potentially put in KEAP1, delete KEAP1, but potentially also, if that's not enough of an effect in your CAR Ts, potentially also knock out STK11 to create even more of a resistance phenotype. So this is useful information. This genetic information allows us to potentially control cells, not only for predicting cancer response, but potentially modulating our environment, especially as we're more likely to be exposed to radiation when we all colonize Mars. Okay. So you might say, what is the cellular context? How relevant are these mutations? So we conducted this analysis in immortalized bladder cell lines. This is TERT immortalized, so it's a different immortalization process. And we picked SNVs that confer resistance in BS2B. And then you can see here that they tend to land in this quadrant. So if they're more resistant in one cell line, they're more resistant in the other cell line. And this is also true for a lung squamous line that has p53 mutations, but no other significant drivers. So that's KEAP1, it's a loss of function gene. How about KRAS? You might think KRAS mutations, gain of function mutations, binary, right? We turn on and off. But KRAS is actually really, really interesting. And this has been spearheaded by Piero Lito at Memorial Sloan. Piero and others have shown that these different alleles actually rewire the metabolome in distinct ways. But we're really interested in the process of KRAS, namely because it tended to confer resistance to radiation mainly at higher doses per fraction. So you can see here the normalized survival much higher at 6 gray than at 2 gray. And so we then took a bunch of KRAS alleles and reintroduced them. So the axis here is y-axis is resistance, positive is resistance, negative is sensitivity. So really interesting, right? So in the switch 2, we tended to see more consistent radiation resistance with perhaps these Q61s giving us the most level of resistance in this particular cell line. D33E is a bona fide activating allele in KRAS. It leads to transformation. Bill Hahn showed this, and so have other groups. However, when you put D33 in, despite the fact that the BS2B cells grow much faster, they actually are quite sensitive to radiation. So that tells you that there's substantial intragenic differences in these important genes, and that, again, binning these into KRAS mutated versus KRAS not mutated is not sufficient information to bring these types of biomarkers to the clinic. We had a cohort that started out in 2012. For those that treat patients with lung cancer know that originally KRAS was profiled, not because we were interested or can target KRAS mutations, but if you had a KRAS mutation, you're unlikely to have an EGFR or other targetable alterations. So it was almost done to rule out the possibility of another targeted gene. So early on in the early days of genetic profiling, KRAS was part of the panel that one used. And so we have this information that goes back to 2012 to 2019. Our panel increased in 2019 to include 18 other genes. And then in 2021, we now have 500 genes that includes KEAP1 and NERV2. So that information isn't available yet, although we are getting that information. But we had a ton of information on KRAS in our SBRT cohort. So we looked, 50% of the patients had biopsy confirmed recurrences, which is actually much higher than the average clinical cohort. So we know that these are true recurrences, and we used rigorous criteria for measuring local failure. And so this is the data. These are the alleles in KRAS that we had in our clinical cohort. You can see this is mainly a reflection of we were initially doing hybrid sequencing of just G12, G13. We really weren't aware or understood very much the switch 2 domain mutation. So this is a feature unfortunately, of how clinical diagnostics was done at the time. So I can't tell you whether the wild type alleles had switched to domain mutations, although those are relatively rare. So the doses at Northwestern University were pretty homogeneous. It's hard. I still don't have a good reason for this historically, because I think those who treat lung SBRT patients know that there should be more heterogeneous dosing, especially for peripheral tumors. So 10 gray per fraction, but this was great for us because dose wasn't really a variable. We can look at the volume as well as the particular mutation. So KRAS mutations aren't bigger. They're actually tend to be a little smaller, although this is not significant. So this is CT axle diameter. So this isn't, the mutation isn't just making tumors bigger, which then confers more resistance. Volume is not a feature in a multi-regression model. And this is the data on mutant KRAS versus wild type KRAS, SBRT. Our local failure data suggests that KRAS mutations are important for local failure after lung SBRT. So you have KEAP1, NERV2, Max's data. You have some data out of MD Anderson that suggests STK11. Our data suggests KRAS. I think I showed you the Venn diagram. There's some overlap between those two. We need more data. Either all of them or some of them, or it's confounded, some genetic alterations, but all these three pathways seem as good candidates for bringing a biomarker into the clinic at the early stages, with the caveat that we need more nuance in terms of mutation profile. Okay, I'm going to tell you really quickly this clinical concept. So we're working with Amgen. Amgen's drug is really cool. It tends to block, so this is sotorasib, it blocks KRAS G12C specifically, although there are other anti-RAS agents that are currently in development that will target all RAS mutations. So we conducted this PDX experiment, KRAS wild type, KRAS G12V, KRAS G12C. You give the drug after radiation, works beautifully. You give the drug before radiation, doesn't work so well. This was very nicely characterized by Piro. So if you give KRAS drug, you block KRAS activity, there's a very rapid adaptive mechanism. You remove the drug for a couple of days before you irradiate. That adaptive mechanism leads to a dramatic increase in KRAS activity. So by the time you radiate, KRAS activity is rebounded, and therefore that's why no effect before the drug. But after, so if you give radiation and then give the drug after for multiple days, you see a really, really good effect. We're going to test this concept with Amgen. Once they have enough sotorasib to go around, we're potentially going to put this in a clinical trial looking at SBRT with adjuvant sotorasib.
Is it present when you give 6 gray when you give it first?
It's not. It's a 48-hour span, right? So if we were to do it concurrently, of course, we wanted to avoid that for the clinical trial concept to give concurrent drug. So we needed a washout phase, but the washout phase led to an increase in the activity of the pathway. All right, so let me tell you briefly about ongoing studies, then I'll kick it to questions. So we're done with, at least on the cell line work, we're done with laborious assays for phenotyping particular mutations, and I think we found another approach, although more limited in scope. I think it's going to give us more information faster. So this is a former postdoc colleague of mine, Alice Berger. She's now at the Fred Hutch. She did phenotypic profiling of multiple variants in lung cancer, 194. It took Alice 18 months to do this. She extracted RNA-seq data using the RNA 1000 from the Broad. So pretty laborious method for phenotyping specific oncogenes in cancer. This took us 24 months to profile variants for radiation resistance. We're going to go to pooled format. So arrayed format is great because there's less noise. It isolates, you know what gene is in which well. You can really get granular estimates of sensitivity. But I think especially if you're going to saturate all of the alleles of these important genes, which is our next goal, we're going to need to go to pool format. And so to do this, we've created this library. This is a KRAS saturation mutagenesis library. You can see the representation. So a saturation mutagenesis library is mutate every allele to every residue to every other amino acid, which is, yeah, it's cool. I mean, people have done this with KRAS. KRAS is a relatively small gene. It's 180 or so residues. So the cost of this library is around $10,000. For KEAP1, it's going to cost us $30,000, which is our next goal. But in addition to, you can obviously put in these mutations, infect them in pooled fashion into your cell of interest, characterize the survivors. You get allelic fractions that increase. Those alleles are more likely associated with resistance, and those that drop out are more likely associated with sensitivity. But we wanted to go a step further. So one of the limitations of single-cell RNA-seq is that none of these alleles are actually captured by the 10x platform or any other platform. So a nuance of transviral gene expression is that there's this huge woodchuck PRE element that is upstream of the poly A site, all right, polyadenylation. So 10x uses poly T to amplify the transcript, but the transcript will just capture the WPRE and won't tell you what variant is in your library. So hopefully I'm not being too wonky. I'm happy to explain this a little bit more, but take it from me. If you infect a cell with a viral transgene, it's not captured in your single cell data, right? They're only capturing the endogenous gene expression. So we want to be able to capture that information because I want to be able to tell I want to do single cell RNA-seq with the mutation of interest in the same cell. So we devised this really cool modification of 10x. 10x is kind of interested in buying this, although I think they're playing hardball. So here's for the bead technology. So you have the poly T. In addition, the bead is attached to. We can attach a 10x barcode with a unique identifier to this trap-seq sequence that we put in our library. So we can actually amplify our gene of interest with a barcode. So every one of these variants that we made in our library has a unique barcode. So we're going to be able to tell you not only in single cell space what the transcript space looks like, but whether it has that particular mutation. And here's a proof of concept. So we did this on a small scale. So this is vector control and this is single cell RNA-seq data, wild type cells. So you can see here, they kind of sit next to the vector control. And then we put in a G12C mutation. So it shifts transcriptomic in UMAP space. And then we put in a really hyperactivating allele of KRAS Q61H. So this is GTPase almost resistant. So once it gets into the GTP bound state, it stays active, continues to fire. And so what's really cool here, you've shifted obviously transcriptional space, but there's still some stragglers. Not every single cell despite this homogeneous expression of the viral transgene, sits in the same space. There's a lot of overlap. So I think we're going to learn a lot about transcriptomics, the particular alleles and what they do to the transcriptome, and then also radiation resistance, all integrated using this saturation approach. PDX, we have a lot of collections, lots of stages of disease that recapitulates. It's not just surgical samples, actually a pretty extensive bronch sample. So lots of stage 4 disease, which I think is cool because the diseases are clearly different based on stage. Ex vivo derivatives that are really highly concordant both genomically and transcriptionally with the PDX and the human tumor. In situ, we've done a lot of this work in small cell, developed close to 70 small cell samples and did a lot of the benchmarking work in that disease. This is the effort, radiofrequency probes into the rough of the neck of the mice. We're measuring radiation sensitivity across these different doses. For some tumors, we've gone up to 16 gray. We've shifted to NRG mice. Some of you maybe this is too much of a detail, but NSG tend to be quite sensitive to radiation. NRG don't have the same DNA repair deficit, and so we're getting substantially better, less toxicity on the skin surface, less ulceration. These NRG mice can tolerate up to 20 gray single fraction to the flank. So if you're interested in doing this, I highly recommend moving towards the NRG space. Some of these patients were directly from SBRT patients, so we're going to be able to do a clinical co-study. So we're going to radiate the PDXs in a manner analogous to the way the patients were radiated. So high dose per fraction, we're going to compare the PDX response to the clinical response. We're super interested in what happens to these tumors after recurrence. So we've harvested a bunch of tumors, especially tumors that have come back late after radiation, to understand genetic evolution of these tumors. We've taken large surgical tumors and we've taken different subregions of those tumors and developed PDXs from them. The idea here is that there's substantial subclonality in larger tumors, and we're seeing dramatic differences in response rates depending on where you took the tumor. And there's really no pattern. It's not that the core is more resistant or the periphery is more resistant. It just depends on where you grab the tumor. And then we're also generating resistance in these PDXs. So those that have been somewhat sensitive, we keep radiating them until we get resistance. Our PDX collection is almost 40% of it has been genetically profiled for RNA-seq and whole exome sequencing. What's a really nice sanity check is that we have very common and very rare mutations that are captured in our data set. So we have a great recapitulation of the genetic diversity of non-small cell lung cancer in this PDX collection, including very rare NRF2 mutations. These are mostly from squamous cancers, canonical mutations like E79Q in our data set. So it's a great resource to also study vignettes of this disease. And then lastly, these are not just tumor cells that are growing in the mice. There's actually a pretty rich immunogenic repertoire in tumor-associated fibroblast repertoire. So when we implant, we take core biopsies and implant them. And from surgical samples, we take tiny pieces. So there's immune cells that stay for 3 to 4 passages within the PDX. So if you conduct cybersort analysis on these, you can see substantial T cell, CD8, CD4, NK cells in some cases. And also a lot of tumors have these tumor-associated fibroblasts. This is mouse. So clearly these aren't just mouse stromal cells. These are human stromal cells that are in these tumors. So one could then begin to tease out intrinsic effects from extrinsic effects, right? Immune cells, different types of immune cells, tumor-associated fibroblasts, sort them out, mix them, determine what's the importance of these extrinsic features on radiation sensitivity, and perhaps answering, I think, this lingering question in the field of what's more important, intrinsic cells or extrinsic cells. I'm going to cut it there. I had some cool stuff on the deep learning side of things that I think we'll potentially talk offline about.
Yes.
And let me just, I was obviously pretty optimistic here. All right, so the takeaway is complex therapy with multiple variants. Binary variants is reductive. I think I've hammered that home. I think bespoke phenotypic assays are necessary. I think the variant prediction tools, I hopefully showed you some examples of where those fail. Intrinsic radiation sensitivity is significant locus heterogeneity. So it's not just one gene at a time. There's multiple genes interacting, and we have to tease that out before bringing this into the clinic. And biomarkers have to be contextualized. There are other genetic events. So if you have an ATM loss of function mutation and a KEAP1 mutation, what do? We need to assess those interactions, right, before you make a clinical decision. And what do you do with a biomarker when you have one? And things that I didn't have a chance to talk to you about. So this is our deep learning models, which I think are ready for prime time. We're about to start a clinical trial using image-based deep learning approaches for personalizing lung SBRT dose. This is our operating system. This is our Vanian's operating system. We're going to be able to modulate dose based on clinical and imaging features. And once the genetic information is ready for prime time, we're going to seamlessly integrate that into that platform. And so that's at least the goals. And don't forget about clinical variables, right? There's other variants in what we do. We tend to think of ourselves as a highly standardized discipline. There's a ton of differences even between my contours today compared to my own contours on the same patient a week from now. And so we really need to automate this process all the way from beginning to end. And you need prospective trials. Don't go too long without running a sanity check. These are our funding sources. The R37 is really what supported most of this in addition to institutional funds. They gave us a ton of money. We're burning through that money on the PDX-ray project. Hopefully they'll give us more. And then we have the UO1 and small cell, which wasn't relevant here. And thank you so much. I'd like to thank people at the lab and thank you so much for your attention.
That will answer questions now. Presentation. Thanks for coming. I've got 2 questions. The first one is this question about the intrinsic versus extrinsic. You know, you presented data on the KEAP NRF2 pathway, SK11, KRAS. It's a pretty big body of data to suggest intrinsic plays a critical role.
Yes.
What are your thoughts on where that data is?
Yeah, I mean, there's a ton of data that KRAS, KEAP1, STK11 are cold tumors, tend not to respond to immunotherapy as well as other subtypes of non-small cell lung cancer. And so I think in that particular circumstance, I think intrinsic is going to be more important than extrinsic. I hesitate because there's some small cohort data that suggests that Nivo improves local control after the Pacific trial, some of that data analysis of Pacific trial and post-hoc analyses. So it's not quite clear to me, but you're absolutely right. These tend to be cold tumors. And so the effects of extrinsic on these particular tumors, I think that's a good bet, but to be determined.
The question was, I think there's really good data for these TEAP1 mutations across multiple studies, including your own, cause intrinsic radiation resistance. If you had a patient with that mutation that was pathogenic, do you think early stage lung cancer would that patient be better served with surgery? Or can you think of trials like that, or do you think the data is premature to kind of think about prospective trials where you're allocating patients based on these pathways? If they're wild type, they should get SBRT.
Yeah, I don't think we're there yet. I get this question in a slightly different form. Are there any tumors that are too resistant that you would not recommend any dose of radiotherapy? There's a small percentage, I would say less than 1% of all the tumors that we've radiated. Some PDXs, even at 16 gray, 20 gray, we can't control. So you have to reduce the volume, and then potentially you can get some local control to get a dose response. I think dose escalation in the SBRT setting is really important. So it's a little bit more nuanced clinical answer to your question. If it's a more central tumor that I'm concerned about dose escalation, but I think I tend to be pretty aggressive in my SBRT dosing, especially with heterogeneous dosing. I think I can control tumors, and if they're not surgical candidates, I have no choice. But as of now, can I a priori determine whether I should not radiate a tumor based on genome alone? I don't think so. And I especially since we don't yet have granular information about the KEAP1 alleles that are the most resistant versus those that are just a little resistant. So I think we're going to get there, but we're going to need more comprehensive genetic data to get there. It also depends on the volume of disease and what the patient can tolerate.
Just following up on that, I know there's lots of questions, but you also told us that intratumoral heterogeneity is important. So does that not conflict with the central conclusion, the importance of intrinsic factors?
Yeah, the intratumoral heterogeneity, it's certainly there's subclonality, and the bigger the tumors, the more subclonality there is. So it suggests there are additional genetic events that are regulating response. What those are, I can't tell you yet, although once we conduct whole genome analysis to really capture those rare events, I think we'll be able to tell you a little bit of what's happening there. I don't think it necessarily conflicts, because I think they're probably genetically driven, and they're unlikely to be microenvironmentally driven, because we're taking these tumors away from their microenvironment of interest and injecting them into a new microenvironment. So I still think they're intrinsic features unless we're capturing specific repertoire. So I think it's early to be able to come to that conclusion, but I do think intratumoral heterogeneity is critical. And you probably saw that slide, the barcode slide, where we label every cell with a unique barcode, expand, and then inject into 8 different mice and radiate the same tumor ostensibly 8 different times to see if the same intratumoral clones emerge every time, to see if survival is stochastic or deterministic within a tumor. So I think it's still an outstanding question.
In the deep sort of allelic profiling correlation with response, are there any non-somatic or germline alleles that you found that are associated?
So we haven't run that assay yet. We haven't infected our KRAS library. There are some germline KRAS alleles that tend to be mildly oncogenic, and some actually block the binding of some of the targeted drugs that are being developed for specific alleles of KRAS.
What about NRF2 or?
Oh, and NRF2 itself. So we haven't saturated NRF2. There's no, to my knowledge, there's no germline NRF2 mutations. There's some SNPs that have been associated with oxidative stress response. This comes a lot from the botany and a lot of Eastern medicine approaches and understanding the SNP associations with particular responses to herbs and other supplements. But to my knowledge, there isn't a strong germline effect for NRF2. Those mutations are almost always somatic.
Thank you for your talk. That was really
Of course.
I was thinking about for the dose heterogeneity and SBRT, we're also seeing much more of that in the oligometastatic states. I'm also quite curious what you're finding in your systematic appraisal of these alterations or these single variations for opportunities to de-escalate, because we're treating closer to multiple organ sites.
Yeah.
And we're not going for the 100 BED equivalents for depending on where we're treating. Just seeing if there's anything coming out. Of course, you mentioned ATM and whatnot, but I wonder if there's something more prevalent that's.
Yeah, I do think by adjusting the dose so that you don't over radiate is going to be really important, not just for primary lung cancer, but for oligomets, especially as we radiate 5, 10 to oligometastatic sites. I think it raises a couple of questions. The polygyny of the metastasis, are they all the same? Will they all respond in a similar way? We have some lymph node primary tumor PDXs. I think we'll be able to answer some of those questions. But in terms of multiple sites of metastasis, as you know, there's different phylogenies, and some metastases come from the same source, others come from different parts of the tumor. So I think that's an interesting question within the same patient, whether you dose every metastasis the same. I think anecdotally, I can tell you that the soil also matters. So when I radiate liver versus lung versus bone, I tend to see, corrected for volume, I tend to see slightly different local control rates, but that's anecdotal. So I think from an oligomet perspective, yes, if you can assume that all polymetastases are created equal, I think potentially this kind of genetic information is useful. However, you'll have to biopsy every metastasis. So I think how do you implement that in a practical way, I think is going to be challenging. Thank you so much.
Well, thank you for everyone today and for Dr. Abazeed. Our next session will be in 2 weeks.